So at EHA 2025 in Milan, I am presenting the IRAKLIA study on behalf of all the co-authors and the Sanofi pharmaceutical company. It's a phase 3 randomized non-inferiority study to demonstrate that the isatuximab subcutaneous formulation with a little device we call an on-body delivery system or on-body injector is non-inferior to the IV formulation. And so in the future, hopefully, will be available for us worldwide. So the study design was a non-inferiority study, phase 3, as I've said, randomized 1-to-1 in patient in a relapse setting using isatuximab pomalidomide dexamethasone regimen. But in one arm, isatuximab was IV. In the other arm, isatuximab was subcutaneous, but not manual push using this on-body delivery system. And there was two co-primary endpoint, two primary endpoints, VGP overall response rate and C trough, the serum concentration, pre-cycle 6. And there was a number of key secondary endpoints, including VGPR rate, C trough, serum concentration, residual serum concentration much earlier, cycle 2. There was also questionnaire, quality of life questionnaires, and other key secondary endpoints. And so the study has 1 year follow-up and had read for the primary endpoints. So two co-primary endpoints, two primary endpoints. The overall response rate, we demonstrated that the overall response rate was very similar across the two arms. And so the study is positive for the response rate. It met the non-inferiority criteria, and the 95% confidence intervals lower boundary was above the non-inferiority lower margin that was qualifying the study as non-inferior, so positive. So that's for response rate, the first of the two primary endpoints. The second primary endpoint was a serum concentration, residual serum concentration, C trough. Right before cycle 6, steady state, per request of the FDA, Food and Drug Administration. And again, it was non-inferior, about equivalent across subQ OBI and IV. It was actually superior in the subQ, but anyway, it met non-inferiority. So that's for the primary endpoint. The two primary endpoints, that were the endpoints that would immediately decide whether the study was positive, and because it's a registrational study, then Sanofi could register, approve, get an approval, and the reimbursement thereafter, or whether the study was negative, and so the study is largely positive. So the clinical significance is absolutely major. It's absolutely groundbreaking for various reasons. Number one, right now, we have two CD38 antibody. We have the historical daratumumab, and we have the isatuximab. It's important to have various options for patients and their families. The thing is that the daratumumab is available as a manual push subcutaneous formulation. The isatuximab is IV. And even with the fast IV 30-minute option, still it's not what the patient wants. What the patient wants, it's something that could not be IV in the vast majority of the time. So here we're going to have a subQ formulation of isatuximab. And so the two CD38 monoclonal antibody, daratumumab and isatuximab, will be subcutaneous, and that's really lovely. That's point number one. Point number two, which is, I think, the most important aspect of the study, is that when I personally, I mean, I have gray hair, so I personally have lots of experience in myeloma, and I've seen a lot of drugs initially developed IV, becoming subcutaneous available, which is lovely and great. But I never thought that we could rethink the complete concept of subcutaneous formulation. And here, this device where you plug your vial, you stick it to your skin, it's entirely automated. It's individualized to the thickness of your skin. The needle is retracted, gets out, gets in. It's really like painless. This way of totally rethinking the subcutaneous formulation with the device is, I think, really gigantically smart. It's so genius. And that is really what I love in the technology, is that we were used to manual push, and we would have thought that the isatuximab would be manual push, but actually it's not. There's a new device, a new way of giving it, much less harmful, much simpler, automated, which gonna simplify the life of the patient, simplify the life of the nurse, simplify the delivering of the drug. And so the fact that the smart people can imagine something totally groundbreaking and genius is really always very interesting. And so this device, I think, is here to stay. This device, I think, it's probably going to be the way for all the sub-Q product to be administered in the future, because the patients in a different study called ISALCO, they said that the device was much better than the manual push. Less pain, less local reaction, much easier to use, much more patient-friendly. So these are the two main aspect of what the study is about. Well, I'm fortunate because I was invited to give the talk and share these incredible developments. I'm happy because it's the technology comes to medicine and helps the medicine, and this is always amazing to see how people very smart can think about improving things constantly, and this is really great. I think it's also great for the patients, for the families, for all the team worldwide, because the CD30 monoclonal antibody are very important drug for myeloma. They have transformed the myeloma survival of the patients, and so we use a lot of them. And so to improve the way we give that drug to the patients is always very important, because it concerns 100% of the patient worldwide. So I do think that this IRAKLIA study is really incredible in that it brings something, I think, amazing to the patients for the future. And this is really, really lovely.